Hi, hello everyone. Hope you can hear me. I'd like to start by really thanking the CRS committee for giving me the chance today of sharing with you some work we've been doing in the Battery Lab. What we're interested in is how artificial and biological networks process sounds. So we're going to start with the biological networks. Now we know that in the mouse, so the model organism we're going to be focusing on today, if you ask this mouse to do a simple auditory task, for example, to discriminate two pure tones, the mouse can do this even if it doesn't have its cortex functioning. But a more complex task, distinguishing a pure tone from a frequency sweep, requires the cortex to be intact. So we know that different sound attributes are extracted at different levels of the auditory system. However, of course, all the information available about sounds is already present at the level of the cochlea. So all that can happen afterwards is a progressive reformatting of this information in order to make it available for perception and the behavior we measure in the mouse. So in the Battery Lab, we embarked on a multi-pronged project to try and document and understand this reformatting at multiple levels. And I'm going to tell you about two features that we saw emerging in our work so far. So first we had to pick the sounds we were going to probe the auditory system with. So we picked a set of 140 sounds that would allow us to cover the basic auditory attributes that people have been studying for a very long time now. Of course, sound frequency and intensity, but also temporal modulations of sound, either in the frequency domain or the intensity domain. So we looked at the responses to these 140 sounds in multiple regions of the auditory system. Here is our data set as it stands today. We have a detailed biophysical model of the cochlear nerve, so I'm starting at the bottom of the screen here. Then moving up through the system, we have a mixture of deconvolved calcium signals and electrophysiology in the colliculus and the thalamus. And in the cortex, for the moment, we just have calcium signals. We're working hard on the electrophysiology. So I just put a sort of overview of the data here to point out three things. One, we really endeavored, particularly with the calcium imaging, to get a really exhaustive sample of the activity in each area to get a really good idea of the sound representations. The second point is we recovered the expected properties that people have already seen in the literature, for example, tonotopic organization. And finally, I'd like to point out that calcium imaging is sometimes seen as slow, but here we're using a deconvolved signal and sounds that vary quite slowly in time. So in the last column, you see a 3 hertz amplitude modulated tone, and you can see that with the calcium imaging, we can really track the sequential changes in our sounds. So once we had the data set, we needed to find a way of fairly comparing representations at each level. So this was a bit of a challenge because since we were combining different methods, we had different dimensionalities, different magnitudes, and sometimes we're using spikes, sometimes we're using fluorescence, and of course different levels of noise because measurement noise is very variable, as you can see on the right of the screen. So we chose to use correlation, that is robust to dimensionality and magnitude, and also because we could devise a noise-corrected correlation measure to get rid of the problem of noise. So concretely what we do, on the left of the screen here, is we take the population responses to two sounds, we calculate our noise-corrected correlation between these two population vectors, and then just iterate this process for all the pairs of sounds to build up a correlation matrix that represents the relations between sounds and their level of decorrelation in each area. And this is what it looks like. Here you see these matrices from the cochlea all the way up to the cortex. So the way to read these is that red indicates correlated sounds, so sounds are going to be hard to disambiguate based on neural data. Yellow is decorrelated, so sounds that are easier to separate. Now, to construct these matrices, I took into account the full sequential activity of neurons in response to sounds. But of course, there's another way of looking at the data, that is to average out time and simply focus on which neurons are firing for which sounds. So here we're looking at a pure identity code. And here I hope you can appreciate, just sort of looking at the general color of these matrices, that there's a bigger evolution using this type of coding between the cochlea and the cortex. So if we quantify this, first by doing what I just asked you to do, by sort of looking at the overall color, looking at the average correlation, we see, so on the graph on the left, the light gray curve that represents the full sequence activity, we see that, decorrelation happens quite early in the system, between the cochlea and the colliculus. However, if we now look at the mean activity, so averaging out time, looking at this time-independent identity code, we see that the decorrelation happens in the dark gray curve more progressively, and it's only in the cortex that the decorrelation reaches a similar level as we see with the full sequence. So we can back this up with linear decoding, and if you focus on the red bar on the far right, so the cortex, it's only here that we see that we have as much information in the identity code as the sequence code. And it's not just about the quantity of information, it's also about the structure of information. Now, if we compare the structure of the matrices I showed you on the previous slide and measure how similar they are, we see that in the cortex they're essentially identical. So what we see is a decorrelation of sound representation throughout the auditory system, but the crucial point we're going to focus on today and the property we're going to try and understand is this appearance of a time-independent identity code in the cortex. Now, I should point out, some of you have probably noticed that something fishy is going on in the thalamus. You see, in fact, a recorrelation here. We'll get back to this later in the talk. But for now, let's focus on this question of time-independent coding. And I'd like to point out that this identity code decorrelation happens for all the kinds of sounds we explored, and particularly for those sounds that I've circled in red that have exactly the same spectral properties and can only be distinguished based on their temporal structure. So, for example, a sound going up in frequency versus the same sound but now going down in frequency. So the only difference between these sounds is their temporal sequence, but in the cortex, they're represented without any reference to the temporal sequence of the neural activity. So why could this be? Why do we see this time-independent representation emerging? Now, we hypothesize that it was linked to the fact that the auditory system is not just describing sounds in time, it's actually categorizing them. And I'll give you a perceptual example of this. So here you heard 3 pure tones that were ramping down in intensity. Now I'm going to let you listen to exactly the same thing, but reversed in time. Now I hope you can appreciate that there was a very different perceptual feel to these tones. You had a sort of a different holistic effect, because you're not just hearing the same sound reversed, you're perhaps hearing what your auditory system is representing, a down-ramping sound versus an up-ramping sound. So if indeed it's true that time-independent representations are linked to categorization, we would expect to see them in CNNs trained to categorize sounds. So we constructed 6-layered CNNs that took as input spectrograms and answered 4 questions about the sound. What is their frequency, their intensity, and how are they modulated both in amplitude and frequency domain? Now the network could do this with a high accuracy, and then we could simply take out the activations and study them like we studied the biological network in response to the same 140 sounds. And we chose either to look at the sequence representation, so preserving time that's inherited from the spectrograms, or averaging out time to look at the identity representation. And now what I'm doing now is putting up on the screen exactly the same figures I showed you before for the biological network. And you see here with the convergence between the light and the dark gray curves that we also see this appearance of the time independence identity code in the later layers. And I should point out that we do not see this in the untrained networks. Now it could be objected that this property is simply a trivial correlate of the fact that convolutional neural networks, because as the C indicates, are performing convolution, the input dimensions, so time and frequency, are actually getting squeezed out of the network to be replaced by more and more channels. So perhaps that this time-independent coding we see is simply due to the fact that time is disappearing in the network and not to categorization. So to disambiguate this, we constructed CNNs, forcing them to preserve the temporal dimension throughout each layer. And if I now show you the same figures you're becoming familiar with for these networks, we also see that we preserve this time-independent identity code in the later layers. So another way to back up our hypothesis is that we should only see this time-independent identity code for sound properties the network actually cares about. Take the example of intensity. Here you have pure tones at 3 different intensities, and we see both in the trained network on the left and the biological system in the middle that we get our time-independent decorrelation. What if now we retrain the network but remove any question referring to intensity? If we do this, we see the network no longer decorrelates sounds of equal intensity. And we can repeat this for the 4 different properties we trained our network on. Now, frequency is a little bit special because it's built into the input dimension, but for the 3 other tasks, we systematically see in black if the network is trained to disambiguate 2 sound attributes, we see decorrelation, but in blue we do not see it if the network was not trained on this sound. So this really reinforces our hypothesis of a link between time-independent coding and categorization. And just as a final check, we thought it would be a good sanity check to take a completely independent network that we hadn't trained to check if we see this property. So we downloaded a network trained by the McDermott lab and kindly provided for everyone that classifies words and musical genres and fed into it the kind of sounds it cares about, either music or words. And what you see here is the now familiar convergence between the 2 gray curves. Once again, we get this time-independent code, now in a very much more ecological task and in a completely different network. So what we started with is the biological data that led us to hypothesize that time-independent representations are linked to categorization. And based on the various analysis of CNNs, we've really backed up our hypothesis. But now, in the last few minutes, let's get back to this thalamic anomaly we pointed out. So when we saw that the thalamus was recorrelating the sound representations, we thought perhaps we need to zoom into our data again from the population level to the single neuron level and go and see what the neurons are doing. And indeed, neurons in the thalamus behave very differently from neurons in the colliculus and the cortex that's around it. Thalamic neurons are very dense. That is, they respond to pretty much any sound you throw at them with different firing rates. This is very different from a typical cortical neuron that is much more picky. It displays sparse coding because it is only going to respond to a few sounds. Now, the literature has already related sparseness and decorrelation, and we could back this up in our data. If we take the thalamic neurons and remove the densest neurons to artificially make the population more sparse, we get decorrelation. So we think that the recorrelation is due to this dense coding, but then of course we just have the next question, which is, well, why is the thalamus using dense coding? Well, maybe it's linked to the architecture of the auditory system. So here you can see the same graph for sparseness on the left, and on the right, the number of neurons for each area. And I hope it sort of pops out at you that these are very similar, and that the thalamus is a very specific region that is a very narrow bottleneck between the colliculus and the cortex. So we wondered, could it be that because the thalamus is a bottleneck, it has to rely on dense coding? So in a hand-wavy fashion, this means that since all the auditory information is being squeezed into very few neurons, all these neurons have to respond to the sounds in order to preserve the information. So once again, we turn to CNNs to test our hypothesis. So we constructed CNNs, picking one layer that we termed the bottleneck, and then removed channels from this bottleneck layer, making it narrower and narrower, and then we simply measured the impact on representation sparseness. Now here is an example. We have a very broad bottleneck. On the left, we get this spotty representation that indicates a sparse coding. But as the bottleneck becomes smaller, here with only 6 channels, we get a denser representation of the sounds. And we can quantify this on the right here. This is sparseness throughout the network, in gray, the bottleneck layer. And as you can see, as the color becomes closer to yellow, the bottleneck is getting smaller, and we're getting denser and denser representations, just like in the thalamus. So we've seen that the biological auditory system is not a simple layer-by-layer smooth progression. We have this very specific effect that we see in the thalamus. And based on CNNs, we suggest that the dense recorrelated representations in the thalamus are due to the fact that it's a bottleneck. However, this work is quite preliminary, and we're still trying to figure out the link between sparseness, area size, and correlation. Now, more broadly, to wrap up what we've talked about today, in this project, we tried to use CNNs to test hypotheses that were driven by the data, but which are very difficult to test in other ways. Very sadly, I don't know how to grow a mouse with a big thalamus or a small thalamus to test my ideas about this effect on representations. But with CNNs, we can manipulate the task choice and the architecture to test how they impact representations and see if it backs up our biological hypothesis. And so on that note, I'd like to warmly thank everyone in the Battery Lab, in particular Jacques Bourg, who did this work with me, and Brice, who supervised all this, and you for your attention.